and welcome everyone. Thank you for joining us today. My name is Sarah Chapman, and I'm representing the Notre Dame Integrated Imaging Facility, as Jill said. Our core is a bit unique as we are essentially 4 different cores under one umbrella. So we house electron microscopy, optical microscopy, histology, and in vivo imaging. And we are very excited about the tools and technology that we're going to hear about in this presentation, because we feel that this is a good linker between each one of those. It fills the gaps where histology, optical, and in vivo might not be able to address. So I'd like to introduce Matt Silva. He is going to be our speaker today, and his colleague Warren is also on the line with us. Matt is the CEO of EMIT Imaging, the leader in Cryo-Fluorescence Tomography. Previously, he served as CEO of InVitro, a global imaging CRO, and led the strategic vision and mission to support the drug discovery and development community with diverse imaging services spanning preclinical and clinical applications. Prior to InVitro, he led imaging biomarker groups at Vertex, Amgen, Millennium, and Takeda Pharmaceuticals. Matt holds a PhD in biomedical engineering from Worcester Polytechnic Institute. And please, Matt, take it away.
Thank you very much, Sarah and Jill, for the intro and the opportunity. Great to see so many people here today. As Sarah introduced, I'm the CEO of EMIT. We're a small company that is developing this novel CFT technology. So what I'd love to do today, and Sarah really set this up well, it's really a bridging technology that provides resolution and sensitivity in large sample volumes, which is relatively unique. And honestly, we created it out of necessity. I'll save that story if there's a question about that. But what I'd really love to do is focus the presentation really in 3 parts. First, I'd love to introduce you to the technology itself. How does it work? I think that's useful when then looking at the applications, because you have a sense of how those images were created. But then I'd like to go back to the instrumentation again and take a little bit of a closer look as to what's going on inside and how do you deal with this really complex, large set of data. So first off, why did we do this in the first place? Well, kind of just recognizing and coming from an imaging biomarker background and a services background, just how often you all and researchers globally need to understand PK, BioD, and delivery systems, and how often you need to understand PD and protein expression. It's absolutely critical in the characterization of disease models, in transgenic animal characterization, and developing new drugs and understanding biomarkers. But you're often left with a choice between either looking at small tissue volumes or large volumes, and trading off things like resolution and sensitivity. And as we move towards more targeted medicines, we're also seeing that we don't often know all the tissues that might be involved in the biodistribution or the disease, or those that are on and off target. And so we recognized an opportunity for us to create we saw an opportunity to create a technology that would address these issues. And I'm going to go into more detail about the images that you're seeing here in a moment. But the point I'd like to sort of lead with is, you're seeing whole mouse. We can do multiple mice at a time, or whole rat. We can do large animal tissue samples imaged at high resolution. So we image between 20 and 55 microns at high sensitivity, so single-digit nanomolars. And what we think that this does is effectively bridges the gap between pathology measurements and in vivo tools. Going through very quickly, and this is illustrated on the right-hand side here about this sort of gap-filling opportunity. We really view this as being a mesoscopic imaging. It's just above cellular at 20 microns, but well below what we're used to in the in vivo animal imaging world, which can be a couple of millimeters of spatial resolution. But there's opportunity for all of these tools to sit side by side. As we look at what CFT offers, it's high sensitivity, high resolution. So where you would tend to look at nuclear medicine for sensitivity, but then look at MRI for resolution, sort of balances the two offerings of those. Offers the ability to quantify. What's really important that you're going to see throughout this presentation is the ability to multiplex. And this is just inherent in fluorescence in general. So IHC leverages this every day, whereas this can be very difficult for other imaging modalities to look at multiple tracers at the same time. PET, for example, this is very challenging. And from an ease of use perspective, I'll show you how you run these scans. It's very easy for users. So it's something that could fit into a core facility in quite an easy way. The reason that this is all possible is due to the instrumentation that we've developed. And again, we're going to go into this in a couple of levels throughout the presentation. But essentially, XerraTM, which is the instrument that we manufacture at EMIT, automates this workflow, sectioning samples, doing all of the imaging, creating anatomical white light images and fluorescent images at high sensitivity, at multiple different magnifications. And with sort of multiplexing capabilities with multiple lasers and emission filters. And as we go through the presentation, the advantages of this flexibility built into the automated platform will become apparent. I think it's also important to note that it's a CE marked instrument, so you know that it's something that is reliably built and tested and robust. So generally, what is CFT? So Cryo-Fluorescence Tomography. Essentially, it's serial block face imaging. So you take a sample of tissue or a whole animal or multiple animals. We embed them in OCT, which is what you would routinely use for cryosectioning of tissue. And then within the XerraTM, there's a series of images. So you saw some flashing. That's it, the system acquiring images. Now you're seeing a blade come and taking a very thin section of tissue away, and then it's going to repeat the imaging, and it will repeat this process as many times, so 800, 1,000, 1,500 times, as many as it takes to get all the way through the sample. So just to be clear, this is a end of life, end of experiment procedure. The animals are in fact very dead. They are sectioned, but this is, truth be told, this is the only way to achieve the resolution and sensitivity that we're seeing in the images. And so the output is what you can see on the right-hand side, an overlay of anatomical images. So these are the photographs, the white light images of anatomy, coupled with the fluorescent images. And I'm going to go into this experiment in just a little bit more detail right now. This is an experiment we ran a couple of years ago as a proof of concept, where mice were injected with AAV9 that drove expression of GFP. At 15 days following IV injection, the animals were exactly as I just described, frozen, embedded, and submitted to CFT for imaging. On the left, you can see a control animal where there is no signal. We're going to talk a little bit about autofluorescence today. In this case, you can't avoid it, right? This is biologically there. This isn't something we have a magical trick against. We've simply just thresholded above the autofluorescence in this particular case. What you see in the panel next to it, what's the fly-through is showing you is all of those areas where there's AAV-mediated protein expression or GFP. Would be surprised nobody here that the liver is the dominant signal. We know that. But what is most important, if you look at the rotating MIP next to that, are all of those areas of green, all of those areas where protein is being expressed, which may have something to do with the AAV's function, either on or off target when you're developing a therapy. There's a couple of zoom ins here where you can see we can identify clearly DRGs. This is where Sarah would also want to take this and confirm this by histology. But what we're providing here is a large view, a whole sample view of where expression occurs. And we think the application opportunity here is pretty broad. So we have groups that are looking at drug distribution, those that are scanning screening delivery systems like LNPs. If you modify an LNP, where does it go? What does it do? There are groups that are looking at this, as I just showed you, whole body protein expression. I'll show you an example of multiplexing drug with, say, cancer cells and looking for co-localization. Looking at on and off target tissues and characterizing them further by biochemical or histological methods. So guiding tox path, and sitting alongside in vivo imaging. So if you want to do optical imaging for the first 20 days of your study, no problem. You're just going to submit the animal for CFT at the end. And this works broadly across therapeutic areas as well. So first example, relatively straightforward. A collaborator of ours called Ratio Therapeutics is developing FAP targeting compounds. For the imagers in the audience, this will be very well known to you. There's a lot of interest here from both diagnostic and therapeutic purposes, radiotherapy. And so biodistribution is really important because that's going to dictate your dosimetry, et cetera. So what they've done is they've fluorescently labeled their drug, injected it into the tail vein of animals, 4 hours later sacked the animals, and submitted them for CFT. And so this panel of images, again, gives you an idea of what the instrument will provide. Whole body image of the fluorescent drug biodistribution on the left, showed in 3D and rotating, and you can do ROIs or whatever you want here. You get the anatomical localization. What is really powerful here, and I'll show you at the end, is in creating ROIs. You're not guessing where the tumor is, but rather you go back and forth between what the anatomy is showing you and what the signal is showing you by fluorescence. And so delineating the liver from the spleen, from the kidney, it all becomes relatively easy because you can just see them. It makes it much easier. In this case, what's interesting about FAP is in juvenile mice, FAP is expressed in the joints. So in the CT image, you can see the bones, you can see the joints, and this is expected, this is on target. And if we zoom in, just to give you a sense of at 35 microns, what would it look like? What would your image look like? So this is a 35 micron section, zoomed in, didn't do really a lot of image processing here, just showing you the raw data. The white light image, so you can clearly identify the bones, the head of the femur here, the joint that where it articulates, and then on the fluorescent image, you can see this punctate light signal of FAP. Really important, other groups have identified this before, gone to RNA-seq to figure out what are these cell types, and then ultimately, what are the implications if you're going to start developing therapeutics. So beyond just looking at your target tumor tissue, what else is happening in the body? We largely believe that this is a complementary method. It's going to fit in with your histology and your in vivo imaging. But like any technique, it has its advantages even over the existing tools that we've come to know and love. For example, in vivo fluorescence, ubiquitous. Everybody has a couple of these boxes. They're easy to do. It takes a couple of minutes. But there are a lot of challenges with light in biological tissue. And so it's really difficult to know if you've got a large but dim object or a deep and bright object because there's so much scatter that occurs with light. And in this particular case, if you look at the image in the, here in the fluorescence image, clearly we can see the tumor. It would appear that the spine has about the same uptake as the tumor. And if you look at the CFT or at the nuke-med image, you can see that this clearly isn't the case. It's just that there's no fat. There's nothing to attenuate that signal before it gets to the camera. SPECT is a great tool, nuclear medicine, coming from in vitro, we did a lot of nuclear medicine. The upside here is the translatability and the sensitivity. So you can take these methods and bring them to the clinic. The downside is it has limited resolution, so 1.5 millimeters plus. It has limited anatomical information. The CT in a mouse largely is skeletal. It's really not showing you a lot of contrast in soft tissue, and it does require radioactivity. So facilities like yours, that's maybe not a big issue, but actually for a lot of groups in pharma and biotech, it's just a big lift to get radioactivity kind of studies off the ground. And so, CFT plugs in there by offering, good anatomical information, high resolution and sensitivity and ability to multiplex. and also this sort of ease of adopting it into a workflow. A couple of quick examples. Here's a study side by side where we look at bioluminescence and CFT together. Again, I think these are good complements. Both have their limitations, right? We're an ex vivo tool, and bioluminescence has, as I mentioned, this light scatter and attenuation issue associated with it. But in this case, you have 4T1 cells on the flank of animals, monitored over time by bioluminescence. So, most folks here who are imagers understand, we've engineered the cells to express firefly luciferase. You inject the substrate, you get really clear images of where the tumors are. And you also get some hints here about heterogeneity within those tumors. So they're just not uniform blobs of signals, but you can see that there's some bright spots and heterogeneity in the signal. That heterogeneity is confirmed by CFT, visualization of the complexity of the tumor. So you can see it floating around. It's the bright blob on the side here. I'm not showing the anatomical images, but you can go to the white light images and see viable and necrotic regions, right? So they appear different in the photograph. But what else here is pretty interesting is these metastatic lesions in the lungs of the animals, which you're unable to detect by bioluminescence, again, because of depth and fur and just the overall signal intensity here is low. So you can see we really blew up the intensity on these flank tumors, which are quite bright, so that we could visualize the signals that are in the lungs. So a really nice dynamic range, and the system is highly linear. So it's really, really good for detecting a broad range of intensities. Another application that we think is really important is as you're monitoring your animal models and you've got constitutively expressing cell lines, in this case, rather than a DS red and bioluminescent sort of firefly line, this is a TD tomato ovarian line that's orthotopically injected into animals. And you can see the side by side of the in vivo and the CFT image. This group also wanted to look at localization with a protein drug. So a monoclonal targeting HER2 that's also labeled with Alexa 647, injected IV into the animals. So we're showing these in two separate panels, but they're all acquired from the same sections at the same time. So they're intrinsically overlaid with each other. So you could look at concordance and discordance of your signals. And then also, for those who do in vivo imaging, you'll see the kidneys here float around in the image. And so you can look not only at your distribution to target, but also your clearance kinetics associated with it. There are groups exploring this in a variety of different ways. I'll give a couple of other examples. Here's one that was nice from the group at University of Wisconsin, who are studying in situ vaccines in animal models. And they have an M-cherry mouse where the T cells express M-cherry. And what they wanted to do as a part of their study is understand what happens to the T cells after animals are irradiated and subjected to IL-2 therapy. So on the right-hand side, you can see this is just a 3D image of the mouse, just the fluorescence, no white light. But you can see the localization of T cells in a normal mouse. So you can see some lymph nodes throughout the body. You can see it up in the sort of nasal turbinates of the animal. So there's a few, Peyer's patches, a few other places where we're seeing some T cells, but none in the tumor. So this flank tumor is cold, and there's no immune cells here resident in it. Following radiation treatment and IL-2 treatment, you can see that there's a substantial change in the animals. So these lymph nodes now are much larger. There's much more signal here. You can see that that's not only local, but there is an increased amount of activity throughout the body, but primarily local to where the tumor is. And then also you can see that the tumor itself is no longer cold. So the ability to sort of visualize from an immunotherapy perspective, what this group could also do is have the B78 cells engineered to express a different fluorophore, so that you could start to look at localization of the tumor cells with those T cells. Just to be clear, it's not single cell level resolution, right? So we're still looking at 20 plus microns, but you're getting a global view and perspective that then, your team, XerraTM, can then submit for immunohistochemistry to better understand. But this helps guide where you want to have a look. Just a quick example of a group looking at labeled cells labeled with Q dots, just to give a sense of like, well, what are some small structures we're able to detect? These are 1.2 cubic millimeter cell pellets sitting in the spleen and liver of mice. So it's a technology, and certainly for universities and researchers such as yourself, offers a lot of flexibility to do some pretty diverse research. One of the areas we're most excited about, where we think we can have the biggest impact, is gene and cell therapy. It's really important, and the FDA just put out some guidance in May on biodistribution assessment of gene therapy products. It's a really complex and hugely growing space. But the question is, well, where do these products go when you inject them, and what do they do when they get there? Where are proteins produced? The vaccines that we all had shot in our arms is relatively easy, right? Like, we know where that went. It's right in that intramuscular space, and we assume, but the distribution to other sites is limited. That being said, it would really be great to know, like, what does happen in the heart? What does happen in other organs? Is there any redistribution or secondary distribution that occurs? But we were doing a lot of work with groups that were focusing on things like ASO. Just want to have a quick look. How do we study skin cancers? Great question. I didn't include an example on skin specifically, but we are working with groups, one in particular that has an antibody that targets skin. And we can, we can measure dermal thickness changes associated with the disease, et cetera. The way we prep the animals is they're fully intact. So there's no perfusion, no fixation, no decal, no nothing, just freezing and go. I probably didn't give you an adequate answer there, so let's circle back in a moment on that one. But what you're looking at here is intrathecal administration of an antisense oligonucleotide, right? So what do you want to know? You want to know that from your injection site, how much of it gets to your target organ, the brain. You're going to want to know, okay, well, how does it clear? Where does it go? And over what time frame? The necessity with CFT is that you would have to take animals down at different time points. But that, the benefit is you're going to get sensitivity and resolution that you just otherwise can't achieve. So on the slide on the left-hand side, you can see that these animals were also tracked by 2D fluorescence. Standard fashion, we all would put the animals prone and supine, so you can try your best to mitigate sort of signal as a function of depth and losing signal. But in this case, you can clearly see here that the upper cervical is being missed simply because the anatomical sort of that positioning of the spine and how that changes sort of actually through thoracic up into cervical. So you just can't see that. So you might misinterpret that as being signal loss here, whereas the CFT reveals, you've got full penetration up the spinal column, and then clearance into the lymph system and into the late nasal terminates, et cetera. So we really see this as a complementary tool. Another example of this with an ASO, here this group was injecting ASO directly into the eye in a transgenic animal, for which there is ASO-dependent GFP expression. So the ASO is labeled with Sci5, so they can see where it goes. And then you have ASO-dependent GFP expression, so taking advantage of that multiplex. In the in vivo image, it's relatively easy to detect this at day 1. Signal is pretty much gone by day 15 or hard to detect. The GFP channel with ASO-dependent GFP is quite frankly relatively useless, because there's just so much there's so much autofluorescence that occurs that you just can't see through the skin. The advantage of our technique is because we are, and to the question of skin, we're sectioning through. So sure, you can see on the right-hand side, there's autofluorescence that comes from the skin, but it doesn't impact our ability to detect this ASO-dependent GFP expression in the eye, and it is co-localized where the Cy5 ASO resides. And it's not in the untreated eye, which is on the other side. So autofluorescence is, perhaps nuisance. We think there's some valuable information in that channel for certain studies, but it's something that we can largely mitigate against because of our ability to localize and because we're physically sectioning. One more example here in the gene therapy space, a group called REGENXBIO, who is developing AAV7 and AAV9 specific drugs, was interested in exploring the differences in the biodistribution and the expression of protein. And so they ran studies. So you can see is in this study, they elected to do the heads separately from the body. So that's why they're in separate panels. They first ran a study where they have AAV7 driving GFP or AAV9 driving GFP, but they also ran a study where they inoculated the animals both with AAV7 and AAV9 at the same time, with 7 expressing GFP and 9 expressing TD tomato. So ability to discriminate the two in the same animal over time. And if you look at the image, red is purely tomato, green is where you're seeing more of AAV7, and yellow, because of just that color combination, is where you're seeing more of the joint signal between the two. If you focus then on panel C, just looking at the leg of the animal and at the bicep muscle specifically, you're able to sort of visualize that AAV9 does a better job of protein expression in the muscle as compared to AAV7. So the first set of image shows the fluorescence overlaid with the white light. The image to the side is just the fluorescence. And this isn't powered enough to look at correlation between PCR. But please note that in this group of animals, they recapitulated the same finding by PCR. So basically, higher copy number in the AAV9 in that muscle rather than the AAV7. So a lot more work to be done in this space to compare CFT for its ability to quantify and how it correlates with gold standard methodologies. But the data, here certainly aligns and is very encouraging. One of the most useful aspects of this, as I alluded to earlier, is you all may not know what tissues to pull to do PCR or to do histology. And so here's an example of AAV9 injected IV and protein expression in the head of a mouse. And you can see there's signal in the tongue. You can see up here, if you can see my mouth, there's signal in the teeth, signal in nasal turbinates. And I suspect that you're not routinely pulling teeth to try to figure out what's going on there. It's painful to do so. Anyway, what really this, the Hungarian glands, there were a bunch of tissues that they identified that they subsequently went on to confirm by immunohistochemistry and standard methodologies. So we find this to be a really powerful technology, not to replace doing those methods, but really to fine tune what you would otherwise do and hopefully prioritize better. It's a lot of work to pull all these tissues, decal them, section them, and stain them. So to get a prioritized list, I think is, my opinion would be super useful for you all. My favorite part of the study, though, was they elected to inject AAV9 into the intracerebral ventricular space. So it was ejected directly into the brain, and they wanted to understand, well, what is protein expression look like then throughout the body? And so this sort of spinning image here, and hopefully you're not all getting nauseous with spinning mice, but this spinning MIP here is showing you protein expression in the peripheral nervous system of the animal following that injection into the brain. So I just think, just as an imaging scientist, it's a fascinating image. Being able to see, looking at the kind of nerve endings that you're seeing in the paws, knowing how sensitive that is to animals, and the ulnar and radial nerves, the DRGs here pop in the spine. There's a really rich piece of information. Please excuse something that's semi-anecdotal, but we gave this in a webinar, and someone from pharma called me later because there is this gap in signal here in the thoracic space, right? There appears to be just really strong cervical signal down into the lumbar sacral area, really strong protein expression, but there's this gap here. And the reason they called is because they had noted this by histology and had never actually seen this whole connected image of protein expression, but they had basically the same finding. So this tool is really helping to visualize biology in 3D, and that's one of our basic premises, right? Biology, pharmacology, all of this occurs in 3D, and if we could see more, we will do far better research. Just one other example here before I get back to just talking about the instrument and how it works. We often get asked about neuroscience and how can this perform in neuro. And the truth is we've done a lot of neuro work. Here, for example, is just a commercially available animal from JAX, where there are a subset of GABA neurons that express GFP. And we at EMIT took this study on ourselves because we wanted to understand what really can CFT provide here. So I'm only showing you a single section, but we get a full 3D view. You can get a sense of, at 20 microns, what does a mouse brain look like? And what does the corresponding fluorescence image look like? We can clearly identify structures. They map very well to the Allen Atlas. We're not here to compete with light sheet. There are super resolution techniques that are available. There's clearing technologies. There are all sorts of great tools in the neuroscience space. We think that there's a great role for us to play here in biodistribution and volumetrics. And we think we, again, sit alongside those other technologies in a great way. The other big advantage we have is ability to look at large tissues. So if anyone in the audience is working in large animals, just a couple of examples. Eyes are very, very difficult to work with for many groups. Here again, just frozen cow eye in OCT, nothing special, sectioned, and here we just image for GFP. We do have some examples where we've looked at AAV therapy before in non-human primates. We've done human eye before as a part of a study. So, we really don't have a size limitation. Here's an example of a pig heart, again, just GFP. An example of a cow brain, again, white light and GFP. So for those of you that are looking at large animals, here is now an opportunity for you to be able to study that. We also do have some groups that are exploring applications with human or clinical tissues. We can share a publication from the group at MD Anderson. They're taking whole cadaver breast and part of the chest wall, embedding the whole thing, again, no decal, just sectioning and imaging that whole sample to better characterize the anatomy and to understand disease better. So it offers a lot of flexibility here. Everything from, there aren't a lot of instruments that can go from a mouse brain to a cow brain and offer a variety of opportunities here for imaging. So that segues back to Xerra, right? So what is it? Well, let me take a quick look at chat, make sure, how can you delete autofluorescence? Great. Let me, before I talk about the instrument, let's talk about autofluorescence. That's a really nice question. Autofluorescence can't be deleted. It just exists, and it will vary by channel. So there will be some autofluorescence you'll find a little bit in each channel. Green tends to be the most prominent. So, what we do for that is we strongly recommend that you put the animals on low fluorescing chow so that you're able to mitigate that. And then we also recommend that, it just won't work amazingly maybe for GI applications unless you have a really strong signal. So this example here is, well, actually that's near IR. I can show you an example here of what a green looks like in a mouse brain. So this is mouse head with GFP expressing cells. This is work out of from Lopez Bertoni out of Johns Hopkins. They've injected a small cell pellet here. My arrow got a little bit misaligned. You can kind of see it on the white light image here. There's a little dark patch. Here are the GFP cells. And then the rest of what you're seeing here is autofluorescence. And I didn't subtract it out because I wanted you to be able to see what it looks like. The upside here is that as long as you have signal that's above it, you can threshold. We're also working on better characterizing GFP so that we can, in a more automated fashion, help users to identify what is GFP versus, or what the GFP, the autofluorescent contribution is to signal. The other question that you asked, just to get that one, how can we prepare tagging molecules? Really great question. There are a variety of methods. There are some kits where you can just do, some basic amine kind of reactions. That's a conversation we're happy to have with you. We have some chemists that we consult with. We're not a chemistry shop per se, but there are many publications on how to label molecules for this. Great. So moving on, what is Xerra? What's on the inside of it? Let me check my time. Okay, what's on the inside? So we've got a cooling system. So the system operates at -15°C. So we keep everything frozen. You got a sample loaded. We've got a blade that does the cutting, and sitting up on top of that is the optics tower. It can move up and down so that you get different magnifications. And as I mentioned, I'll talk in a moment here about lasers and filters. Continue. Before we do that, let's just talk about prepping samples. It's relatively simple. Frozen sample, embedded in OCT, mounted on one of our stages. The stage is loaded into the system. The tower is replaced. The user selects what are your fluorophores, blah, 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 put in a study name, all that you need. And then the system will do the rest. It will do the imaging and it will do the sectioning. And depending on how your universities like to handle data, we push everything to the cloud and do all our processing there. But everybody's different. So we would work with you to figure out what works for you and your IT departments. In terms of sample sort of flexibility that you have, we have 5 magnifications and hence 5 block sizes. So you can put isolated tissue in the field of view A and get 20 micron pixel size. So a comment on pixel size and thickness. We come from the in vivo world, and so we tend to think isotropically. So our pixels are 20 by 20 microns, so we section at 20 microns. Field of view E, large block where you're looking at 22 by 15 centimeters. You're putting 4 or 5 mice, whole rats, large samples. Our pixel size is 55 microns. We section at 55 microns. That's just kind of the bias of where we come from, and it's easier from a processing perspective to assume that everything's isotropic. But you can see here that you've got a variety of options. And I will tell you that there are groups, including us, exploring, well, why don't we just not lay the mice that way? Why don't we lay the mice kind of up and down? You'll acquire way more sections. We've done that. We've done whole mice at 20 microns. And you can put a couple of mice in one of the smaller blocks, just positioning them vertically. For lasers and filters, we tried to cover the range that matched really biological applications and what we believe you all would do. We continue to optimize lasers for performance in most common fluorophores, but you can see that we go from green to near IR. We cover very common Alexa dyes, etc. On the right-hand side here, you can see how we've characterized those dyes to look at limit of detection and quantification. These are all nanomolar numbers here. So we continue to test more. We're continuing to understand the performance of the system. The other important point is we do this by a dilution series. So we'll put a dilution of fluorophore in the system, and linearity of the system across exposure times and across from the dilutions are all above 0.99. So it's a highly linear system. What Xerra does is automate acquisition and data processing. So the pre-processing to do all the warps and the dark field and flat field corrections. And then from an analysis perspective, yes, we're building our own tools, but quite honestly, many groups like Sarah's group, I expect, has 5 tools that they already use. So we try to be as flexible as possible in our data formatting so that you can use those. One of the main problem with existing tools is, they're just not used to opening a 2000 by 2000 by 1000 image. A lot of them get really grumpy. Oddly, ImageJ and Fiji do a great job with big images. But that's something that, we need to continue to work on. But I mentioned earlier, one of the real beauties here, and I'll wrap up quickly here in the next minute so we can take more questions. One of the beauties is having this molecular image sitting alongside an anatomical image. So there's not a lot of guesswork as to what an organ is, because you can just zoom in and see what it is. And you can see in this is just an example of me doing some analysis. But the mouse kidney is, relatively big on a CFT, right? You can really zoom in and see it. And so identifying the pelvis from the medulla, from the cortex, relatively simple. Or here in this tumor, I segmented it into 2 regions because if you actually look, you can see that they separate. But your ability to identify structure and then determine the amount of signal coming from that structure, quite frankly, having done imaging for a long time, it's pretty easy here compared to some of the other tools that we use. So just wrapping it up, and hopefully I didn't inundate you entirely with examples and information, but we're excited to show every picture we acquire, to be quite honest. But hopefully I shared with you a new and different way, right? So this is a new modality, as Sarah introduced. It fits into an existing gap, and we really wanted to achieve high spatial resolution that complemented existing tools, but that were differentiated from them. High sensitivity, so we knew we wanted to detect low concentration of fluorophore. If you're labeling a drug, you don't want to just see, you don't want to see physiological. You want to see pharmacologically relevant doses is the best way to say it. Something that's biologically active and not just an enormous dose so you can see it. Relatively high throughput. It takes about 15 to 25 hours to go through a block. So most groups that are using this in a high throughput mode, you can probably get 20-ish animals done a week. So at the resolution and sensitivity, high throughput, I think, is maybe relative to other techniques. But for the amount of information you get, it's really extraordinary. Complements what you're doing, simplified workflow that's not very challenging. You don't have to do a lot of animal processing, and that can be multiplexed. So you can look at multiple molecular signatures at the same time. I will end it there. Be very happy to take either to jump back into other questions if I didn't adequately answer them, or take any new questions that you might have.
So Scott Snyder, I'm a pet radiochemistry director here, Matt. A couple of quick practical questions, actually. So if you're slicing through an entire mouse, and you're going to do this overnight, apparently, because it's a 20-hour process, does the blade last through the entire mouse, or do you start getting bad cuts partway through? First question. And the second one is it possible at all to capture any of the slices to then go back and do like immunohistochemistry staining or something like that? Or do they get trashed as they get cut?
So both fantastic questions, and every time I present, I think I got to put that section collection slide in here. Yeah. And you'll see why when I answer, you'll see why it's a little bit of a dicey one. But all right, first the blade question. Yes, it's a specially designed blade that is intended to go through a whole block on its own. We do recommend you change it for each block. And sometimes, like if we're doing a pretty dense sample, so we've got some non-human primate heads coming up, we'll probably change the block once, the blade once or twice in that run, maybe once. But it's intended to last the whole time. And you can just remote in, depending on your IT privilege. You can remote in. There's cameras in the system. You can verify that everything looks okay, and you're on your way. You will see on some of the images that sometimes you'll see little streaks. It is not uncommon for a bit of bone to chip and then push through the remainder of the slice, and then the next slice is usually fine. So there are some sort of physical limitations and issues of, we've talked about grinding and doing other things to get absolutely flat surfaces, but our hesitation is because of your second question. There are groups that want to collect sections. So the way that you do that, and the reason we don't have a slide yet is we've not really perfected it, but it's with a tape collection. So if you're familiar with what you would do for autoradiography, you pause the system during its run, you sort of open it up, you put a piece of tape and adhere that to the surface, and then as it's sectioning, you pull that tape up, and then you can take that sample and bring it over for IHC. We're going to have more information on this. Our engineering team is doing a mini sprint on really improving this blade speed, blade angle, and actually blade type. So we are contemplating the idea of swapping in more of a histology blade for one or two sections, and then switching back to our blade. We're just concerned about, like, do users want slices bad enough that they're willing to kind of go through all that? Some of our users, to maybe a third way to answer this, we have a really diverse set of users. Some are academic groups that really want those samples. Others are more like pharma groups where honestly, they just want to get through as many animals as possible. They want to triage their compounds, and everything is just spent at the end. They throw it away, and they'd rather have a satellite animal at high quality rather than a CFT section at moderate quality. Was that too much, or did that answer your question?
No, that was perfect. Thanks.
Great. Any others? Please speak up. Happy to answer. And I will say, if there are no questions, clearly I did an amazing job of making it clear. But we're also not going anywhere. So we're highly committed to the technology, hoping to work with you all. You can email me, Matt.Silva, or you can email Warren.Davis, or EMIT. And we're more than happy to talk about your specific research and how CFT can work, because as a new technology, we recognize it can be, it's not like an MRI where you're like, okay, I know what an MRI does. Can you tell me about your gradients and your bore size and that kind of stuff? We're introducing sort of a new way to do things. Scott, I think you said you're on the chemistry side.
Right.
So you're doing a lot of radio work, which I love. It's absolutely fantastic. We published with Ratio at EAM really high correlations between CFT, quantification, and PET. It's an area we still need to explore more. And we just think that the two methods can sit side by side. One offers translatability, the other offers resolution. So we see them as fitting together. The other, hopefully the engineers won't be upset with me. We are having a look at whether or not we can do Schrank-off in the system too, so that we can support radiotherapeutics. It's really a question of either how dark can we make it, or how thick a blanket can we throw over it. But that work is that's close to my heart. I had published some stuff there, so we're taking a look there because we think there's an opportunity to support that community as well.
Thank you again for the presentation. It was really a nice overview of what people can start to think about for their own research. And so we thank you for presenting at our seminar series, and have a nice day, everyone.